I'm going to actually talk more about the motivation and the sort of theoretical background to doing systematic genetic approaches in functional genomics, and not the nuts and bolts. And then you'll hear right afterwards from Max really how one implements this with CRISPR-based screens and can do this in sort of a massively parallel way. But what I'd really like to do, as I said, is to talk more broadly about what type of systematic genetic approaches have been tried, have been done, what has worked in different systems, and what's starting to work now in mammalian systems. So really focusing to begin with on yeast, because so much of what's now being done was worked out in yeast where there's very nice results. And the other thing is, please interrupt me at any time. I intentionally made my spoken part of the lecture less than the hour and a half allotted so that we can have more of a dialogue. And this is particularly important because I know people come from very different backgrounds, so what would be obvious to some set of people won't be to others. So with that, the outline of my talk is, I start naturally enough with an introduction, and then really talk about systematic genetics and lessons from yeast, and these ideas of using arrayed screens, using deletion libraries, and then introduce what has, I think, been a particularly powerful approach of combining classic epistasis analysis between genes with systematic approaches. And this is a tool for mapping out gene function, and talk about a few of the successes in that approach. And then I'm going to go now to mammalian systems, introduce different approaches for doing systematic genetic analysis in mammalian cells, in particular haploid genetics, very briefly on shRNA, or RNAi-based approaches, and then much more time on CRISPR-based gene modulation, and talk about applications to gene target ID identification, synthetic lethality in cancer, and then also mapping out something we call non-oncogene addictions in cancer. So with that, I'll start and say, are there any questions before I jump in? So just as a backdrop, when the Human Genome Project was started in 1990, or started to be proposed in the early 90s, the cost per base of sequencing was $10. So a 3 billion base pair genome, like yours and mine, would have been a $30 billion project, sort of almost on the scale of the moonshot, and arguably much more important scientifically. But clearly at $30 billion per genome, we weren't going to do a lot of genomes, not a lot of mammalian genomes at least. By the time the first drafts of the human genomes came out, the cost per base pair had gone to 10 cents or so. So then it was going to be $300 million per full genome. Expensive for sure, still, but a pretty dramatic drop of an order of magnitude in 10 years, almost on the scale of Moore's Law. Since then, we've gone from a $300 million base genome to more or less at this point being there at a $1,000 genome. And to give you an idea of the scale of this, if automobile manufacturing had undergone a similar increase in productivity and efficiency, a $50,000 BMW would cost about a quarter. So it's completely unprecedented, and it's super exponential, so faster than Moore's Law. I think we now are probably back down to going to be Moore's Law, because at this point, the cost of storing and processing the data is getting to be a substantial cost of the overall cost of sequencing. And we're still going to be then limited by how fast computational improvements are made. But it's almost at the point where sequencing has no longer become limiting. And the consequence of this is that we now can sequence any individual's or any organism's genomes rapidly and at low cost. And we can watch the RNAs that are being expressed. We can look at the rates of different proteins that are being translated, or the set of epigenetic marks, and watch these as you go through an interesting biological or disease process. And this has been a remarkable boon to biology, of course, because we have this huge amount of information. But it then has just brought this enormous bottleneck of how do we take this vast streams of data and turn them into some meaning about the function and the functional significance of this. And that's really been the motivation behind having these types of functional genomic tools that let us ask what is the role of each protein and what is the consequence of changing the levels of these proteins. And of course, for now, the last 150 years or so, the basic strategy for understanding what the normal function of a gene is has been genetics, and that's basically strategies for perturbing those genes, either by random mutagenesis or other alternative approaches, and seeing what happens to the organisms. And this, of course, all started in a serious way with Mendel and looking at wrinkled or yellow or green peas. And the insight of Mendel was, of course, the Mendelian genetics, the idea that there was specific inheritance patterns when you crossed a mutant and a wild type, this idea that you'd have a recessive or a dominant mutation. And the reason, of course, that was so fundamental is it implied at that time, knowing nothing about the method of storage of information or inheritance, it implied that there were specific physical loci or genes that were responsible for these traits, and that you had two copies of these genes in the plants in this case, so that you could understand the inheritance by whether one or two copies of the mutant gene were passed on to the progeny. And of course, we eventually, starting in the 40s and 50s, identified nucleic acids as being the information that was encoding these genes, and understand that mutations, changes in nucleic acid sequence of the chromosomes, were underlying the phenotypes that we see. And then eventually techniques became, molecular biology techniques, starting in the 70s and 80s, became available that we could clone, as we say. Basically, that meant we could identify the molecular basis of these changes and then understand the genes and their functions and the proteins that they encoded. And this is really, that led to a real revolution in genetics, because instead of being just sort of this conceptual inheritance of traits, you now understood the molecular basis of these traits, and you understood that they were due to specific changes, typically in the expression level or the nature or function of individual proteins, and therefore had an understanding of really, in many examples in quite exquisite detail, what was the molecular basis of both normal traits and disease. The problem with these approaches, the classic genetic approaches, though, was that you had to rely on getting a mutation in genes and knowing what phenotypes, what behavior to look for. And so it was a great way to identify the first set of genes in a process that you were interested in, but very hard to reach any sort of saturation, and also really didn't scale that well. It was hard. You ended up having a bottleneck of identifying or cloning the genes that were responsible for it. And although these types of random mutagenesis and then phenotypic screening still are a mainstay in molecular biology, they have largely, as the tools have become better, been started to be replaced by systematic genetic approaches. And really, so how many people have heard of reverse genetics? Right. So basically, the forward genetics is looking for a phenotype, finding a mutation that gives you this, let's say one that gives you a wrinkled P, and then identifying the gene that's responsible. And a reverse genetics is basically starting with a gene or a protein coding sequence, altering it, and asking what happens to the phenotype. So instead of going from the phenotype to the gene, you go from the gene to its phenotype. And what's really made these types of reverse genetics approaches so powerful is having genomic sequences and having tools for making systematic collections of mutations. And so I'm going to talk to begin with about the work in yeast doing this, and then we'll hear about how really much more powerful approaches, I think, of pooled approaches for Max later on. So this, as I said, really started with the budding yeast Saccharomyces cerevisiae, which, as I'm sure many of you know, is the organism responsible for fermenting grapes to make wine and for allowing bread to rise. So an organism we've been taking advantage of for several thousand years at least. And it was also the first freestanding organism whose genome sequence was determined. It was published in 1996, and it was a paper. It was just when I started at UCSF, and a great paper, a commentary that came along with the publication of the sequence was Life with 6,000 Genes. And what we learned is that there were really only 6,000 genes, which I think had been estimated fairly well, but was kind of a stark to see all of them. In yeast, there's very little alternative splicing, so it's really just 6,000 protein products. There's a few examples here and there that contradict that, but for the most part, that's what we had. We knew that quickly that about 1,000 of those were essential for viability under standard growth conditions, and the other 5,000 you could delete, and the cells would maybe grow a little more slowly, but basically do more or less okay. So in other words, we weren't going to learn a lot by just looking at the deletions. All we were going to do is be able to bin the genes as being essential or non-essential. Probably the very first thing if you're going to separate genomes in terms of some level of importance. But what made yeast such a wonderful system is its genetics, is that it exists most of the time, at least in the laboratory, probably not in the real world, most of the time as a haploid. So a haploid means there's only one copy of each gene. So if you knock it out, there's no second copy that you have to knock out. But you can cross two haploids, make a diploid, undergo a process of sporulation, and those genes will then reassort themselves into four tetrads, or four haploid strains, each of which any particular gene will segregate in a so-called 2-to-2 manner. The classic Mendelian manner, which means that if you have an A and an alpha mate, when you sporulate, you'll get two. For any gene, you get 2 copies from the A and 2 copies from the alpha. And what the other thing about yeast that was so great is that you could, by very simple, it had terrific homologous recombination. By very simple PCR-based strategies, you can insert in an antibiotic or an oxytrophic marker that would replace any given gene, and do this in a way that scaled readily. So really quite soon after we had the first genome, we had a library of deletion mutants. And those libraries were 5,000 genes instead of 6,000, because for 1,000 of those we couldn't get the haploid. So there's now strategies for making compromised alleles of the 1,000 essential genes. But basically, we could get alleles for each of the individual genes, and then these could be arrayed in a 96 or a 380 well format, where each well had a different gene deleted. And the scale of the yeast genome being only 6,000 genes meant that if you put this in 384 well plates, you could fit an entire genome collection into 15 different plates. So it meant that one could then grow by sort of simple robotics and pinning, grow your entire collection under many different conditions and try to see which genes are important for which types of processes. So, very quickly, if you were interested, for example, in the process of protein synthesis and translation, a simple approach for getting at which subsets of genes you should be focusing on for translation was to grow on a series of sublethal concentrations of translation inhibitors. So for example, cyclohexamide or translation inhibitors that work at different steps in the process of protein synthesis. And that then developed a list of genes that were cyclohexamide sensitive. And so if one was interested in the process of translation, you can now get to this subset of genes and start looking at these in much more detail. A few years later, actually, Erin O'Shea, when she was at UCSF in my group, then developed libraries where we made a GFP fusion to each of the different genes and did microscopy to look at the localization of each of their corresponding protein products. And with that, you can now, if you were interested, say, in the process of protein folding in the endoplasmic reticulum or anything that's going on in the endoplasmic reticulum, take the 400 proteins that are localized to the ER, and work with those gene deletions and study them in depth. And it really was a very big change in the way of sort of thinking about how to identify components in a process. Because before the genome sequencing, when you were doing genetics, it seemed like there was sort of an infinite possibility and you were trying to find the few genes that might be interesting for the process that you're studying. But afterwards, when you start to now have at most 6,000 genes, but when you were looking at the ER, only 400, so what you could fit on a single plate, if you had anything that that organelle was doing that you were interested in, whether it's secretion or folding or putting on sugars or disulfide bonds, it was just a matter of figuring out what the contribution of each of those 400 genes were. So you could do fairly intense studies on each of these 400. You can certainly trivially run gels to look at the effects of folding of different ER proteins. So it really made the world in many ways a much smaller place when you're starting to search for function. And it made the process of identifying the functions of these different proteins much faster. So are there any sort of questions about this? And give you the background. All right. So what quickly happened is for many of the well-studied processes, you found it was possible to identify what a protein was doing or how it contributed to that. But soon, again, shortly after this, by 2002, so only 6 years after the publication of the first genome, Charlie Boone, working at the University of Toronto, developed an approach that was sort of the next step in implementing, going from this classic genetic approaches to systematic genetic approaches. And what Charlie did was to develop an approach that would let you look at gene interactions. So how much the function of one gene was affecting the function of a second one. And to do this on a very large scale. And again, as a backdrop, these types of genetic interactions were classically used. So before people could even clone a gene, if you had a process and you had two genes that were contributing, say, to signal transduction, a classic way of trying to order them was so-called epistasis analysis, where you asked, well, if one was upstream of the second one, if you lost the upstream gene, there should be no function for the second gene. And also a very important complement to this was the so-called synthetic lethal analysis. And that's essentially looking for combinations where each individual gene doesn't have a big impact on growth or on a phenotype. But then when the deletions are combined, they have a much stronger effect. So quantitatively, so now that we have 6,000 individual deletions, or 5,000 are viable, in principle, it would be straightforward to make all combinations and to look at what the phenotype of the double mutants are compared to the singles. The numbers, as you probably start to realize, get big quickly. So if you have 5,000 genes, you have 5,000 times 5,000 combinations, or divide by 2. So 12.5 million, because A, B, and BA are the same, but 12.5 million combinations. And if you have 20,000 genes like you and I do, that's 200 million different combinations. But the sort of remarkable thing is by robotics, as I'll go into in a little bit, it was possible to really measure a large fraction of all the double mutants. But really, to start with, the question was, so once it was clear that there would be value in doing this type of systematic genetic interaction analysis, I think there now have been a number of different ways of implementing it. And so I think Charlie's approaches, I think, were really powerful, both because of the actual data they gave, and as I'll go over briefly, many very interesting biological stories have come from this, but also just because I think it serves as a really nice motivation for doing this in higher eukaryotes. And I'm going to talk about much more, when we get to mammalians, I'll talk about much more practical, disease-oriented types of applications. So with that all as a background, the idea of the genetic interactions are really quite simple. You have a wild type cell, and in this case, we're measuring fitness, so how fast it doubles. And we set that arbitrarily to 1. And we look at the consequence of deleting gene A or deleting gene B alone on this growth, or in combination. And when we combine these two mutations, for most genes, we'll see that the double mutant will be a little bit sicker than either of the two singles. And actually, the vast majority of genes, it's a multiplicative answer. So if you have a 90% growth rate when you delete gene A and a 90% growth when you delete gene B, when you combine them, their growth rate is 0.9 times 0.9, or 81%. And really very nicely, because we've now done many, many of these doubles, that's true over a wide range of different processes. That's when the genes have nothing to do with each other. But in a small subset, you'll have double mutants where we call it positive or buffering, where the double mutant's healthier than you would expect based on the individuals. Or alternatively, ones where they're much sicker, and we call those negative or synergistic interactions, the so-called synthetic lethality, where the double mutant is much sicker than you would have predicted. And when these happen, it implies something very specific about the functional relationship between these two. So anyone want to, give an example of why you might get a positive interaction? Just the type of processes that would go on in a cell that would lead to two proteins. What's that? Yeah, so I was thinking of something in a way just really simpler, which is,
If there's one opposite, if one is more inhibitory and one is more what promotes something, so obviously if you kind of put them together, they kind of actually combine and give you an increase rather than a decrease.
that's possible. There's really something just very more simple, which is if A and B were in a binary complex, so it's an AB. So if A and B were in a binary complex, then if you were to delete A, B isn't functioning at all. And if you delete B, A isn't functioning at all. So the consequence of deleting A or B is the same. But once you've lost A, there's going to be no effect of losing B because B is not working anyway. That's really sort of the classic thing where you think of there being positive interactions. Negative feedback loops or these types of things that people are mentioning can give you true suppression, where examples where when you delete one gene, cells are sick, but when you delete a second gene, it makes the cells healthier. So that's true suppression. And that's very interesting, but implies something different about the function. How about synthetic lethal, where you can lose either A or B, and the cells pretty much get along okay, but you can't lose them both? Yeah.
Kind of redundancy.
Right. So that'd be the classic example would be either true redundancy, where there happens to be two proteins that are more or less doing the same thing, or effective redundancy, like the fact that yeast can grow, they can make ATP by either fermenting, how they make alcohol, or by respiring. And if you knock out fermentation, they do actually pretty well as long as they have lots of oxygen to respire. And if you knock out respiration, as long as they have a fermentable carbon source, they do quite well. But if you knock out both, they're not going to live under any circumstances because they don't have any way of making ATP. So just more sort of conceptually or diagrammatically is that we think of positive interactions often as being a linear pathway or binary complexes, where losing A or losing B has the same consequence, which is you lose the product. So once you've lost A, there's no effect of losing B, and vice versa. And the negative or synergistic is when you have two different processes that contribute to some essential function so that you can limp by without either A or B, at least in the laboratory, but you can't survive at all without. And I bring this up just because these rare cases where you get these genetic interactions or static interactions are indicative of very specific functional relationships between the genes. And but as we can get this in a very large scale, these types of genetic interactions, we can now learn about proteins which are working together either in this sort of synergistic or parallel manner without having to know anything about what those genes or proteins are doing or what the process that they're affecting is. So it lets us sort of start to put this set of 6,000 proteins into these types of functional networks, and to do this in a way that is really sort of free of the standard biases of trying to understand, DNA replication or chromosome segregation, or trying to pigeonhole genes into particular functions. So it's been a very powerful approach for identifying really new biological processes that either because we didn't have the imagination or because of a historic accident, biologists had not previously been interested in. And that, I think, in a way, is really the most fundamental and transformative thing that's happened with the systematic genetics. So much of the systematic genetics was doing what cell biologists did previously, like studying secretion or DNA replication, but just doing it much faster, being able to identify the important genes much more quickly. But beyond that, with the systematic genetics, we should start ask more broad questions about how related different processes are to each other, how many genes did one gene tend to have a genetic interaction with, and what are the different, what are the full set of roles of different genes? So I'll go into a little bit, again, the granularity of how this was done, because partly it's a neat story, and also I think you can see that it was a very nice approach for a yeast, but it was not an approach that was going to work for mammalian systems. So the idea behind Boone's system, the so-called SGA system, was that you could take this collection of deletions, strains, and you would have one collection knocked out with one antibiotic resistance marker and a second with another in a different mating type. So you'd have gene A knocked out with one marker in an A cell, and then gene B in an alpha cell, mate them, you get a diploid that would have both antibiotic markers, sporulate them so that these genes randomly segregate so that you'll get some that are double mutants, some that are single of the two, and then some that come out as wild type. And as long as they're not linked, they'll come in a very, the standard predictable ratios. And through some very clever selection strategies, which I'm happy to offline talk about, but I don't think are really important for this discussion, it was possible in a systematic way to select only those double mutants and only the double mutants that had gone through this process of mating and sporulating. And so now with this, you could generate the double mutants and study, for example, how fast they grew or whether they were more or less sensitive to a drug than either of the two single mutants. But as I said, there's huge numbers here, so how could you start to approach the 12 million? And the answer to that was basically just simple robotics. So they developed a robot that had 1,500 or so colonies on each plate and pinning it in an automated way. And so this is actually the picture of, here's the so-called plate hotel that feeds into this robot that then crosses, takes the 1,500 alpha and 1,500 A's and crosses them together, puts it on another plate to select the diploid, put it on another plate to select the double mutant haploid, and then let that grow and take pictures of it to see how fast the colonies were growing. And so, you can imagine if you wanted to do, a million with 1,500 per plate, it's not so many plates, but actually you have to go through multiple plates to do this selection, and you have to do it in high number of replicates to get good data out. So it was a massive effort to get sort of the 5 million types of genetic interactions. And that was terrific and has been a huge boon to science, especially to yeast genetics, but it wasn't going to be repeated under 500 different conditions so quickly. But it really, but I think really a lot of the value was that it allowed us now to see what the potential of these types of genetic interaction maps were. And I'm going to try to go from, I hope you can understand why looking at genetic interactions, you can start to get at the functional relationship, like if they're redundant, then you get synthetic lethality. But I'm going to change gear to what was actually, I think, has proven to be the more valuable readout that has been enabled by this type of approach. And that is, it was essentially an automated way for any gene to get a very rich phenotype, and a phenotype that would often be very specific to that process. So the problem with a classic genetic, with the looking at growth, for example, is that it would just categorize genes into two different groups, the essentials and the non-essentials. So you say maybe how essential they were. But essential genes could have almost no relationship to each other. One could be in translation, the other one could be in DNA synthesis. Similarly, genes that cause a mild growth could be in very different processes. So it wasn't rich enough to tell you what that gene was doing. So, and what you got then out of these large-scale genetic interaction maps is for every gene, when you deleted it, what its pattern of genetic interactions were against, in principle, the 5,000 other non-essential genes. And here's an example of one particular gene, and you can see that it happens to have a buffering interaction with UBC4 and a synthetic interaction with RPA14. And a lot of other different genetic interactions with different genes. And even if we knew nothing about what A did or why it was buffering with UBC4 or why it was synthetic with RPA14, we could then take this pattern and compare it to other genes and say, do these patterns look the same? So in this case, right? They're quite different, and you can quantify the difference between these patterns of genetic interactions. And that would tell you, with a high degree of resolution, what the consequence of losing these two genes were. So what then, if we had this type of complex AB, what would we expect for the pattern of deletion of A versus the pattern of deletion of B? Similar. Right, because the genetic interactions, these patterns of genetic interactions, are just reading out on the total loss of this complex. And it doesn't matter in this to first approximation which way we broke these complexes. And this has proven to be true for a wide variety of different complexes. And what would it, when we know all the physical complexes, which is very nice data on pull downs on who's interacting with who in yeast, what would it tell us then if they had different phenotypes? I'll give you one specific example. Actually, it was one that we used. What would you hypothesize if, when you looked, A and B are interacting, you knew that from a pull down. If you saw that the pattern that A had was anti-correlated, was the opposite of what the pattern that B was, what might you hypothesize? Right, exactly. So the example that we had, just to bring this to a more concrete thing, was there was a gene that was identified in a GWAS study as being very important in childhood asthma. It had a yeast homolog. There was essentially no functional data on either the yeast or the human protein, so we were starting from scratch, really a cold case. But what we knew was this ORM by pull downs was interacting with the sphingolipid biosynthesis pathway, and the first enzyme in this. And what we got from the genetics was that the loss of the enzymes that were making these lipids had the opposite pattern of the loss of this ORM interacting partner. And what we were then able to, with a lot of work, to infer and to really show is that ORM was an inhibitor of the sphingolipid biosynthesis. So that when you lost the enzymatic enzymes, you had less sphingolipids. When you lost ORM, you had more sphingolipids. And so you got this opposite phenotypes. And it's that idea that by having this sort of systematic genetics, we can paint on the physical structures in the cell, these AB complexes that we can also measure in large scale, the functional relationship between them, that I think has been one of the things that's sort of most powerful as a combination of understanding the physicals and the functional organization of proteins in the cell. So here's an example of an actual map. This one is focused on protein folding in the ER. Each line here is a gene, and you're not supposed to read what the genes are because that's not the important point. What we've done here is to simply reorganize the genes by which ones have similar patterns of genetic interactions. So you can see these sets of genes have this similar pattern of buffering, of aggravating interactions with this set, and they end up having very similar function. And similarly, you can see these set have the same pattern of positive interactions. What was the nice thing about this and other similar studies is that it recapitulated a lot of what we knew about ER biology, or in general about cell biology. And that's comforting because there's been a lot of people doing beautiful cell biology in yeast. In fact, there's more people working on this organism than there are genes in this organism. So we know a fair bit about what the different proteins, most of the proteins, are doing. But despite this, there were highly conserved, and there still are, although dwindling numbers, highly conserved proteins from yeast to man that have essentially completely unstudied. And what was nice about this map and other maps by various groups is that in addition to making these tight associations, these similar patterns of genetic interactions between proteins whose function we knew, there were also many examples where there would be a tight correlation between the genetic interaction patterns between either completely unknown genes, or in this case, between 3 genes, one of which we knew exactly what it did. The other 2 were either partially or completely uncharacterized genes. So in this case, we saw these 2 proteins that were uncharacterized. One was so uncharacterized no one had even bothered to give it a name. It's called YOR164C, which just means it was a yeast on ORF on chromosome R, and it was the 164th gene on the CRISPR side of that chromosome. And it immediately then gave a hypothesis that this would be important in this process of so-called tail linker insertion, and that proved with actually fairly straightforward experiments to be the case. And again, I'll jump into just a little bit of detail here because I think it illustrates how one can use these types of maps to get at function. So we knew that the GET3 protein was important for inserting a class of membrane proteins, so-called tail-linkered membrane proteins, into the membranes of the endoplasmic reticulum. So these are proteins that have a C-terminal transmembrane domain and are anchored in the ER or other membranes with their cytosolic, most of the rest of the protein in the cytosol. And GET3 bound these tail anchors. It bound to a receptor called GET1, GET2, and in an ATP-dependent manner, inserted it into the membrane. But one could infer that this was unlikely to be the whole process because proteins are going to be synthesized on the ribosome, and this tail anchor is going to be the last thing that's going to be made. So it was unclear how one would get from these, from the synthesis of the protein to onto the GET3 complex with this would then insert it. And these new components that had the same phenotype as GET3, one of them was a ribosome-associated component, and as is turned out, and now it's worked out in a number of labs in very elegant detail, these are really there sitting on the ribosome, ready to catch the protein as soon as it comes out, and then to transfer it to the GET3, and then transfer it into the membrane. So it makes sense as this was one single pathway that they would have a similar set of genetic interactions. And incidentally, the reason these 2 hadn't been identified previously is all the studies that had been on the GET system had really focused on things that were actually at the ER, so physically associated with the ER. So it was great at identifying those components, but not so good at identifying the upstream components. And I gave this, first of all, is there any questions on this particular example or how we would go from these types of maps to inferring a functional relationship? Great. So now this is another list which is only meant to illustrate that these are focusing on the ER and things were done in my lab, but similar studies have come out from many other groups, including people working with Charlie Boone and Nevin Krogan, my colleague at UCSF, where in focusing on different parts of biology, it was possible using these types of genetic interaction maps as a start to basically shoehorn yourself into a new piece of biology and quickly find out the functional components and their functional relationship between them. And in sort of what's the ultimate complement for these types of functional genomics studies, that by the time these things came out, the actual screen or the genetic interaction maps were a detail at most in sort of figure 1, and the rest of it, the strength of the papers were really on their biological insight rather than on the novel methodology. I think, and really stepping back, my point is that this is really has become a proven way, a very rich approach for getting at, in a systematic way, getting at the function of genes, and one that is a very nice complement to other large-scale types of functional studies, like where is the gene localized and who is it interacting with. And by having these data sets in yeast in a systematic way, where for any gene you can ask, where's the protein localized, who is it genetically interacting, who's it physically interacting with, it puts people in a position where if you're interested in a biological process, you can do an awful lot of the initial research just by going through the various databases. And in a way, it's put, I should say that the sort of, in some sense, I think there was a certain resistance in classic cell biologists for these types of systematic approaches as being seemingly mindless. But I think that's actually, I think that's a minority view now. And I think it's a really sort of misguided view, because rather than sort of taking out the insight, I think it actually now makes it it lets people who have the insight, who have an interesting biological process, or something that the cell is doing, and a new insight into what the cell processes that should be important for the life of the cell or a disease process. Now you can, instead of having to do months of work or more to start to test this out experimentally, you can go through these available databases and start to generate hypotheses before you get your hands wet. So now, I think we're in the enviable position that we're sort of more limited by ideas and imagination of what the cell is doing than we are by the sort of raw data to at least get started. So with that sort of part proselytizing and giving you background, I'm going to switch gears and spend the second half of the talk talking more broadly about how similar approaches are being implemented in mammalian cells. And we're going to hear, so you've heard a little bit from Luke about these approaches, and we're going to hear in much more depth from Max about how these are really implemented in experimental strategies. But I think I'll, by way of motivation of this, I think just in the last year or two, we now have these tools in mammalian cells where it's really possible to do things on the same sort of scale that we've done in yeast, and in many ways now actually faster and better in mammalian cells. So if we've had the genome since 2001, why is it that I'm saying only in the last couple of years? What are the challenges for doing this type of similar genetic approaches in mammalian cells? Yeah, so that is the key challenge in doing genetics in a mammalian cell, is that you have two copies of the gene. If you do any sort of either directed or random mutagenesis, it's going to be hard to knock out both copies and only those two copies. So there's really conceptually two ways to get around this, assuming we're not going to actually be able to specifically knock out both copies, which now we actually, with CRISPR cutting, is suddenly become feasible. The two approaches are, you either find a cell that actually happens to be haploid or near haploid, and as crazy as that might sound, there are such cells, and they've been very useful in making a mammalian cell more yeast-like. The second approach, though, is effectively to build a dominant tool that will knock out or just either knock out or destroy the function of both copies of a protein. And those two tools have been originally RNAi-based and now more recently CRISPR-based tools. So one of the reasons why these CRISPR, either CRISPR cutting or CRISPR-I tools or A tools have taken off so quickly is because a lot has been done with RNAi-based approaches for doing this. So I'm not going to talk a lot about RNAi. I'm sure you've all heard about it, if not used it. So how many people actually have used RNAi? Yeah. So very nice way, if you have a specific gene, you make a double-stranded RNA against it, either short for mammalian or longer for non-mammalian systems, and you take advantage of this endogenous machinery that recognizes double-stranded RNAs and through a specific and intricate process leads to digestion of mRNAs that have copies of these double-stranded RNAs. So what are, I put it up there, the challenges? So first is getting a good knockdown. And then, of course, the second is making sure that you're knocking down that gene and only that gene. And there's a lot of technology between RNAi. There's lots of checks that let you give an idea whether you're getting this type of specificity. But I would say, having had a fair bit of experience in this, these let you use it as a tool, but they don't really get around the fundamental problem of this efficacy and specificity and this balance between efficacy and specificity, because the better you knock down your on-target gene, the more likely you are to also have a broader range of off-target effects. And this has really limited their use in this type of systematic approaches, especially when you start to think about doubles, where if you're knocking down the gene you intended and then 4 other genes, and with each of your 2, when you do the double, you get the AB pair that you care about plus 24 other pairs that were not the pairs that you were caring about. And fundamentally, I think the problem was that you're taking an endogenous machinery that's important for the biology of the cell and trying to hijack to do it something that it wasn't exactly intended to do. And that's really limited our ability to engineer and to change the system to do the things that we want to do. And what has made the CRISPR, whether it's cutting or IRA, I think move so quickly is that it's a simple system, a single protein and a single guide RNA. It's completely bioorthogonal, so there's no endogenous function of the Cas9 system. So we're able to change it at will without worrying that we're changing an important biological process. And so this has given a degree of the ability to do engineering and to tap across the wide bacterial biology of CRISPR to manipulate gene function in a way that's really unprecedented. But let me talk quickly about haploid genetics. So, and I think it's still a very valuable tool. It was developed by Jan Carret and Tijn Brommelkamp. And basically, they took advantage of the fact that there were cancer cell lines that were diploid in the large majority of the chromosomes. You can now do, in this case, they used retroviral gene traps or random insertion that led to stop codons in introns and would destroy the function of proteins. And just give you an idea that you could actually, this can be really done on a large scale. Here's an example of several different proteins where you see when NFATC2 is not targeted, it's gone, and similar for TEC1, et cetera. So you really get very high specificity, clean knockouts, essentially akin to the deletion library that one has in yeast. And by building up a library of these knockouts, you can do this in a systematic way. And this has been particularly useful for identifying, for example, a receptor for a pathogen. If you have a virus and it's using a host protein to bind to the surface and get in, if you then infect your different deletion mutants of this haploid cell line with the virus and look at which cells are able to survive, you can very rapidly identify proteins that are essential for getting into that virus. And this is continuing to be applied to a wide variety different processes. All right, so I talked a little bit about RNAi and a little bit more about the haploid genetics, but now I really want to, naturally enough, we're in a genome editing course, focus on the CRISPR-based approaches. And for the CRISPR-based approaches, there's, as Dana alluded to, there's really two fundamentally different strategies. Both, I think, are proven to be very effective. There's some complementarity, and I think different places where you'd like to apply the different ones. But on the one hand, the approach has been to use the nuclease to make a cut in the gene that's programmed by this guide RNA. This then gets repaired typically by non-homologous end joining. Non-homologous end joining, NHEJ process of DNA repair, tends to cause indels to be formed they tend to be short, but over a random length. When the indels don't have 3 or 6 or 9, aren't going to cause an in-frame deletion, this would cause then nonsense mutation, which would change the frame of the protein, and if this is at the 5' end of the protein, have a very high chance of inactivating. And then making a library of these is just a matter of making a guide RNA or a series of guide RNAs for each of the gene that you want to target. So very much like the haploid genetic approach that I just talked about, but because the nuclease can cut both alleles with high specificity, you can get nulls in even in diploid or even aneuploid cell lines. So effectively, these type of nuclease-based approaches let you really complement the haploid genetics, but to do this in a much broader range of cell types, and even to do this in an in vivo setting in a mouse. The alternate strategy has been to use a catalytically dead Cas9, or dCas9, that was actually developed by Stanley Chi when he was finishing up here as a student and moving over to UCSF as an independent fellow. And he's now he's been migrating slowly south, so he's now down at Stanford as a faculty member, but still a great collaborator on all of this. And with this, instead of having a nuclease that's driven by the RNA sequence, you have a programmable DNA binding protein. This can then be fused to different effector domains. So it can be used, as Stanley initially showed, to block transcription. You can bring in epigenetic modifiers to turn on genes or turn off genes, or to change the genome. So essentially, if you think about genome editing, the catalytically active Cas9 as a great tool for editing the genome, the dCas9 is a tool for editing the epigenome. And what has made this really move so quickly, as I think you heard a little bit from Luke's lecture yesterday, is that there's been all this fundamental groundwork that had been done using things like zinc finger or tail proteins as a way of recruiting different effector domains. So many of the uses and the tricks of the trade had already been worked out and could be, in a really often straightforward manner, be applied to dCas9. Though, of course, the transformative thing about dCas9 is in order to get a new binding to change the specificity, all you had to do was generate a new guide RNA and do it in a predictable and simple manner instead of having to design a new protein. And then, of course, as you also heard, you can fuse this to green fluorescent protein and light up specific loci. Really, for these screening lectures, though, the most important functions of these are to either bring in silencing domains. So typically, we've used a CRAB domain, but other silencing domains have been studied, or an activation domain, like a viral VP16 protein. And then in this way allows you to turn on or off any gene of interest. And the beauty of this is that it's turned out to have a very high degree of specificity. So this is an example where we're targeting a green fluorescent protein that's been inserted into the genome and looking at RNA-seq analysis either in the cell that is expressing Cas9 in the guide against GFP here, or the cell that's not expressing the targeting guide RNA. You can see basically every other endogenous gene has minimal change actually within the noise of the RNA-seq experiment, and this was really quite a clean RNA-seq experiment, whereas GFP is effectively shut down by almost a couple of logs. So now we have sort of these precision tools for turning on and off any gene at will. And what this has allowed us to do, some application of this approach, is basically we have a rheostat. We can define the relationship between the level of expression of that gene and its phenotype. So example here is a gene SD3-Gal4 and sensitivity to a plant toxin called ricin. So each of these is a different guide RNA. On this side is a CRISPR-I guide. On this side is the CRISPR-A, the activation guides. And you can see the different guides give you a range of from no knockdown to 100-fold knockdown. And the activation go from sort of 0 to 10-fold activation. And you can see there's a very nice sort of monotonic relationship between the degree of sensitivity to this toxin and the level of this protein, SD3-Gal4. And you can also see that once you get, in this particular case, once you get to about 90% knockdown, the ricin's phenotype doesn't change anymore. And that means effectively the protein, in this case, the protein is no longer, the residual protein is no longer having much actual function. The sort of most obvious and most, I think, will be broadly used function of this, use of this is to do the type of genome-wide genetic screens that we talked about with yeast in the RAID libraries. But now we can do this for any transcript. So we can get at essential ones by doing partial knockdowns and also get at non-coding transcripts because whether the transcript is functioning as a protein coding protein or as a long non-coding RNA, in terms of this technology, it really doesn't matter. We can use it to shut down the expression of that transcript and look at how it affects the growth or other interesting phenotypes. The CRISPR activation. So why do we want to do activation screens? Why would we want to do high copy? Because that's been a tool, that was something that haploid genetics and RNAi simply couldn't do at all. So anyone run across or seen or used these types of overexpression screens? Have an idea where they might be valuable? Yeah. Yeah, so that's a complement that, absolutely, that it may be very hard when you start to knock down a gene that's essential. The cell might just either not grow or become so sick that it's very hard to make sense of what it's doing. But this lets you do a gain of function. Other reasons? So where do you see natural, in people, when do you have natural examples of overexpression? Yeah. Yeah. So cancers are genomically unstable. If overexpressing a gene increases, say you have a growth factor, and by overexpressing it, you make it constitutively active or broaden its activity, it's a very simple genetic way for a cancer cell to gain its stronger growth phenotype. So when you put cancer under a selection, for example, if you do new targeted therapy, if there's a way for the cell to get around that targeted therapy by simply overexpressing either protein that's disposing of that drug or getting around to the effects of that drug, that will happen rapidly because the copy number of genes, of chromosomes, is unstable. So it's fairly simple to get amplification. In fact, you're constantly getting amplifications in the copy number. So resistance mechanisms or turning a proto-oncogene into an oncogene often occurs. And then the others for redundant genes. And then finally, in the same way that we can do this with a single or double in yeast, we can make, it's conceptually very straightforward and now experimentally, I think, fairly straightforward, at least to do combinations of two and even potentially higher combinations to knock down in a programmed way combinations of genes and to do these types of massively parallel combinations. And I'm going to really talk a bit more about this because it's particularly relevant for thinking about therapeutic strategies, in particular trying to design polytherapies. So places where you really need to target more than one gene to get an effective response. And of course, now has become the classic example of this is in HIV and now HCV therapies, where things like reverse transcriptase inhibitors are effective drugs, but are transient in their activity because the virus is rapidly evolving and quickly gets around any single monotherapy. But by hitting simultaneously multiple processes in the virus, one can take multiple kind of mediocre drugs and get together a cocktail that can effectively prevent growth, if not cure the disease. So I'm going to talk about one other example. And again, I should say, I didn't go through this in the yeast examples, but this is really very well worked out from yeast. And that is using these types of systematic genetic interactions, systematic studies to identify the targets of a drug in an in vivo setting. And this example was to a drug called rigosertib, or Rigo. It's in clinical trials, including late phase stage 3. So those are the big trials that cost a lot of money and involve a lot of patients for myelodysplasic disorder. It seems to cause a G2 to M arrest and apoptosis, and has been implicated in inhibiting at least two different types of kinases, polo-like kinases and PI3 kinases, but other pathways have also been implicated. So now to get an idea, when you treat a transformed cell line with Rigo, what actually is causing the cell death? We carried out genome-wide CRISPR inhibition and CRISPR activation screens against a CML cell line that was treated with several rounds of Rigo pulses at about the IC50 and then allowing recovery. And in summarizing quite a bit of data, each of these is a gene, and what we see is the effect of either overexpressing in the CRISPR-A axis or inhibiting that gene in the CRISPR-I axis, the effect on sensitization. And so most genes have no effect in either screen. In this case, though, it's actually in many ways the CRISPR activation that was the most informative. And the strongest hit was a gene called KIF2C, an alternate kinesin that had been implicated as a destabilizer in microtubules, spindles. So these are, of course, the processes that attach the chromosomes and allow them to segregate to the mother and daughter cells. And here, overexpression of KIF2C sensitized the cells to Rigo, and knockdown of KIF2C conferred resistance. And what Marvin Tannenbaum and Ron Vale's lab noticed was that in addition to KIF2C, several other CRISPR-A hits were microtubule destabilizers, and several of the CRISPR-A hits as resistance mechanisms were stabilizers. And this led to this idea that the thing that the weak link that Rigo was causing cell death, had to do with destabilization of the microtubules. So basically, anything that promoted microtubule destabilization was making the cells more sensitive. This, fortunately, was fairly easy to test. A really nice visual test Marvin did, switching now cell lines, so again, showing that this isn't just true for the specific cell line that we're working on. He worked at a sublethal dose of rigo, and here the spindles were able to form appropriately and attach to the chromosomes during mitosis and allow segregation of the chromosomes. Similarly, the KIF2C overexpression had a relatively modest, if at all, effect on spindle formation, microtubule formation, and attachment to chromosomes and segregation. But when these were combined, it had a devastating effect on the spindles, where they just collapsed. The chromosomes weren't attached, and there was no way the cell could undergo mitosis effectively. So it really gets at the molecular basis of how we think rigo is killing cancer cells, and which then, would suggest some, I think at least in some obvious ways, how one might enhance its effectiveness. And certainly, for example, I wouldn't combine this with Taxol, which has job is to stabilize microtubules, because you would think that they would have a counter effect, whereas other compounds that might cause destabilization would have more of a synergistic effect. And the nice thing is, it would be fairly straightforward if one were making derivatives of rigo and trying, doing so-called structure-activity relationships to look at how, through these screens, at how the activity of your compounds are changing as you change their chemical nature. And I'll talk a little bit more about that in an example in a bit. So just take the remaining 15 minutes or so to talk about a few applications of CRISPR-I and CRISPR-A to cancer biology. I should say, I think CRISPR cutting type screens would also be similarly applicable. And conceptually, we can think of two different types of genetic causes of cancers have been identified. On the one hand is the so-called oncogene mutation. In this case, you get a natural proto-oncogene, an activating mutation in this proto-oncogene to turn into an oncogene. And this oncogene now acts as a driver, forcing the cell to have uncontrolled growth. The second type of mutation are in tumor suppressor genes. These are natural breaks. And here, a loss of function in both copies of the tumor suppressor removes the natural break and allows the cell to undergo uncontrolled growth. So the big difference, of course, between these is the oncogene function is dominant. It doesn't matter that you have one copy of the normal gene. As long as you have the oncogene, it's going to drive tumor formation. Whereas in the case of the tumor suppressor, it's only the homozygous loss, the loss of both copies, that allow growth. And in fact, that's really the basis of how these distinctions were first made, whether you got the mutation was dominant or so-called recessive. And how might one think about treating, how would the nature of these mutations think about change your strategy for thinking about treating or developing therapies? So if you have an oncogene, what would be oncogene mutation? What would be? Right. So I'll show you an example on this slide. You would ideally, you get a mutation that causes an activation of an oncogene that's driving growth. Ideally, you would make a small molecule that only bound to the oncogenic form, the mutant form, so didn't touch any of the non-transformed cells, but inhibited the oncogene. And if this oncogene was a true driver, you can have dramatic responses. And that's really transformed the face of cancer therapy. So that is targeted therapies against driver mutations and oncogenes is the mainstay now of cancer therapies. Here's an example that I think very nicely illustrates both the promise and also, I think, the enormous challenges to these approaches. About half of the melanoma, so melanoma is fortunately one of the rarer but also the most deadly form of skin cancer. It's about 80,000 or so cases a year, and this leads to about 8,000 or 9,000 deaths. About half of the cases of melanoma, there's a specific mutation in a protein called BRAF in the RAS pathway, very well characterized cell growth pathway. And at a given residue, one residue, residue 600, in which valine is replaced with glutamate. This causes a constitutively active form of BRAF inhibitor developed by Plexicon and Genentech, Roche, was made called vemurafenib that specifically inhibits the V600E-driven melanoma, and this can have dramatic effects. So here's a patient pre-treatment and 2 months after treatment. So obviously, a terrible metastatic case of melanoma. Each of these is a separate nodule or locus of the melanoma, and you can see virtually every one of these responded dramatically, so that there's an obvious improvement. In fact, this treatment really led to a change in the way that drugs are approved against these types of targeted therapies are studied in people. When you have these types of dramatic responses, it was essentially argued that it was unethical to do a control arm, because you knew what was going to happen in the control arm, which is the patient was going to get. There was no turning back on this. There's a case in which there were brothers that both had the V600E melanoma mutation, and one was put on the control arm and one was put on the normal arm. And there's a story in the New York Times about this, and it's sort of, I think that and the sort of obviousness of the clinical need really changed the way the FDA thought about approving drugs, so that they would take, when you had dramatic effects responses like these, that they would approve drugs without a control arm based on just historical data. And that's really, of course, what you want. You want a cancer therapy that you don't need to go to do a careful control to get a 20-fold statistical significance that normally that you normally need, one that's all or nothing. So if the story stopped there, it would be a tremendous success, and instead it's a qualified success, because this is a patient 3 months later, and this is maybe an accelerated course, but not atypical, where you see not only has the disease returned, but virtually every one of these nodules has returned. So even though they're not visible here, they were present perhaps in a microscopic way, and each one of these nodules found its own solution around having the V600E mutation, and possibly a different solution, and then was able to grow back. And from this, it's become clear that these types of targeted therapy that we need to begin with to map out what are the escape routes to the targeted therapy, and to block those escape routes ahead of time, rather than allowing the disease to return and then trying to block these. And actually, the V600E-driven melanoma has been one of the test cases for doing multiple therapies up front. So there's now inhibitors and other components of this pathway, in particular a downstream component called MEK, and there's approved double therapy, so that up front the patients are treated with both the V600E against BRAF and the MEK inhibitor. And there's an argument, actually, the MEK inhibitor, that the double treatment, rather than being more toxic, can actually avoid some of the toxicity and side effects of vemurafenib. So I think this is pretty much what I've said before, that the lesson in targeted therapy is we need to map out escape routes if we're going to, if we have a highly rapidly evolving pathogen like a cancer, or as I talked about, like HIV, we need to map out the escape routes and to have block those a priori. And this has led to a search for what has been called so-called non-oncogene addictions. And what I mean by a non-oncogene addiction is a change or a biological process that in and of itself doesn't cause the cancer to grow. So it's not like the V600E. It's not driving transformation. But it's a process that the cell, the transformed cell, the tumor cell, is relying on much more robustly, much more strongly than normal cells are relying on. It's very closely linked, of course, to this idea of synthetic lethality. So what we'd like to do is to understand what are the pathways that a transformed cell like this V600E mutant, what are the biological pathways that the cell is particularly relying on? And one set of such processes that we and others, especially Susan Lindquist, but many others closer to the clinic as well, are interested in the so-called chaperone or proteostasis networks. These are the things that make sure proteins fold properly and that the cell disposes of misfolded proteins. And there's a number of reasons to think that tumors are going to be specifically sensitive to the function of the chaperone or proteostasis networks. First of all, they have a high mutational barrier. So many proteins, both oncogenes and non-oncogenes, are mutated. These mutated proteins are going to have compromised function and more propensity to misfold. They have the wrong number of chromosomes, extra copies of chromosomes, which means that they'll be making proteins and complexes in the wrong ratio. And those uncomplex proteins, so if you have A and B, and A is on a chromosome that's duplicated, but B is on a chromosome that wasn't duplicated, now you're suddenly going to be making too much of A, which can never complex. And tumors are generally in an intrinsically stressed state. This very nice proof of principle of this is that a proteasome, this central protease in the cell, is a targeted, very effective therapy, inhibits the proteasome in melanoma. And there are also several other compounds that are in late stages of investigation, including HSP90 and mTOR inhibitors. The problem, and again, this is one of these busy slides that's only meant to illustrate the fact that there's a lot going on. The problem is that these proteostasis and chaperone and protein degradation pathways are complex and overlapping. And although we've done a lot of very nice work as a field in studying individual components, who they bind to, how they work, how they release their substrates, we're mapping out how these work together is really at the forefront of this work on stress networks. And in particular, I think, good reason to think that the vulnerabilities in a given tumor will depend on the specifics of that tumor type. So whereas the proteasome inhibitor, this garbage can of the cell, is essential for the function of every cell, but you can get very nice specificity and really with relatively little toxicity for multiple myeloma, it's no good at treating the vast majority of other tumors. And we just don't understand really the basis of that specificity. And in the end, we're just going to need an empirical mapping out of which of these pathways are particularly sensitive in which tumor type. And I'll talk a little bit about some work that uses a chemical genetic approach, a combination of small molecule inhibitors plus these types of systematic approaches to try to do this empirical mapping out. This is really work that we've been doing with a colleague of ours at UCSF, Jason Getswicki, who's a spectacular chemical geneticist, has made a series of small molecule inhibitors of a key chaperone, it's called HSP70. This is a protein that's conserved all the way from bacteria to humans. And in humans, there's multiple different copies. Every different organelle where there's folding has another copy of HSP70, and there's in the cytosol, multiple different copies. So it's multiple different specialized forms of HSP70. And, in terms of this mapping out, so there was a number of reasons to think that 70s would be a good target for cancer. But in terms of mapping out, the question is, which 70 orthologs is best and safest to target, and in which cancer? And as a first step in this, Jason looked at a series of small molecule inhibitors of 70. He had two inhibitors in particular that were interesting. This is now up to JG 400 or so. In vitro, these inhibited all the different HSP70s with similar specificity, similar affinity. This first compound, though, they both killed cells. HSP70 is essential, that's not a surprise. They better. This first compound, though, had no specificity for a transformed cell line over a normal fibroblast. So selectivity index of 1. That's by definition a really terrible drug. The second compound, even though in vitro was no different in its in specificity, in vivo it had almost an order of magnitude specificity, and now he's up to 2 orders of magnitude of specificity, where you really start to think this could be a serious specific drug. So the question was, what was it about these 2 compounds when you put them on cells that was changing? So did a screen here again at the IC50 where it's just starting to kill cells and ask what genes, when you knock down, sensitize the cells to this drug. And the idea of this is very simple. When you're working at the IC50, the point where the drug is just starting to have a real effect on the growth, whatever it's hitting, that protein is now limiting for growth by definition. Because if you increase the dose, you'd lower its activity and the cells would grow less well. So if we decrease the levels of that target of the limiting protein under these conditions, we would expect a synergy that's going to make the drug much more effective. And that's essentially this type of screen that was done. And what we saw with the JG84, this is the one that had the 8-fold specificity for a transformed cell, is that one version of HSP70, HSPA9, one that's found in the mitochondria, of course, the power cell source of the cell that has its own protein folding environment. Knocking that made cells exquisitely sensitive to JG84, whereas knocking down the other HSP70s had relatively small effects. Whereas this non-specific compound, JG13, we see knocking down any one of several HSP70s has a mild but clear sensitization effect. So what we learn from this are 2 things. First, satisfyingly, we're pretty sure that in vivo the reason these are killing cells is by hitting HSP70. That may sound obvious, but it's actually far from obvious. There have been many drugs that have been studied, that have been identified based on you take a purified protein and you find a small molecule that inhibits that, and then you put it on the cell and it's really acting through a totally different mechanism. So it's not a wild goose chase. That certainly could have been the case when you saw this discrepancy between what you saw in the test tube and inhibition and what you saw in vivo. But the other thing it said was that knocking down the HSPA9 made these multiple myeloma specifically sensitive to this inhibitor. So among these HSP70s, the one that we should really go after for specificity, at least in this tumor type, is HSPA9. There's also gave us a really nice clue of how this specificity was being achieved. And that's because the difference between 84 and 13 is 84 had a positively charged ionizable group. And so the idea is that even though they inhibit all the HSP70s with similar specificity, because of the proton motive force in the mitochondria, this causes this JG84 to become charged inside this matrix of mitochondria, and that leads it to accumulate inside of the mitochondria. So when you're treating cells with one concentration, on the outside, the concentration of this drug inside in the mitochondria is going to be much higher than the cytoplasm. So now it gives us sort of a foothold for thinking about specificity and how we can achieve this specificity in vivo. So I think I'll just finish then with, I've talked about oncogenes and how mapping out escape routes and these so-called non-oncogene addictions is a strategy that can be enabled by systematic genetic approaches. And then finish now with talking about tumor suppressors. So for oncogenes, we said the natural strategy at first was to target the oncogene for inhibition. But for tumor suppressors, what would one do? Yeah, so many cases, and that's actually been tried. So some mechanism of oncogenesis by tumor suppressors are that you activate a protease that's destroying, say, p53. And if you can inhibit that protease, you'd restore the p53 tumor suppressor. Most cases, though, the gene's gone. There's a big chunk of the DNA missing. You've had loss of heterozygosity. You're not going to resurrect that protein. So there's no direct target. So what do? Give you a hint. It's my favorite trick. So
Induce apoptosis.
certainly you would want to induce apoptosis, but how would you get specificity?
Synthetically started, find a target which synergizes this loss of tumor suppressor.
Right, so that's exactly right. So the presumption is here is that the tumor suppressor was doing something important for the cell, and that when it's lost, there are going to be other genes that the cell now has become addicted to. And so if you can find another gene that's synthetic lethal with the loss of the tumor suppressor, so a gene that on its own doesn't cause much of a defect in growth, but when combined with, say, loss of p53, causes cell death, you now have a legitimate target that will allow you to both be efficacious and to be specific. And so the way you think about this is you have a chair that's got 4 legs, and one of the legs has been broken, so it's a tripod and fine. So your job is to find the second leg to break to make it unstable. The classic and really the only example that I think that will change is BRCA1. So BRCA1, as many of you know, is a gene that's mutated causes susceptibility to breast cancer and ovarian cancer. Typically, affected person, a woman in this case, would have one mutated copy. That would make it much easier by loss of heterozygosity to lose the second copy, and on the second hit, you then are prone to forming tumors because you have not lost a function of BRCA. BRCA1 is involved, as we know, in DNA repair. The reason cells are able to live without BRCA1 and not only live but thrive is that there are alternate processes that are important for DNA repair that are orthogonal, that basically are backup, effectively redundant for BRCA1 function. And a critical one of these is the PARP pathway. So when there's damage, a single DNA break, single-stranded break, that's recognized by this PARP pathway and repaired. If one loses components of the PARP pathway, or more relevantly inhibit the PARP process, this single-strand DNA doesn't occur. When DNA is replicated, it then ends up with a double-stranded break. But this would then be normally restored by HR-mediated DNA repair, and it's this HR-mediated DNA repair that BRCA1 plays a critical role in. So if one both inhibits PARP, so you generate double-stranded breaks, and inhibit this major mechanism for repairing double-stranded breaks, it causes cell death. And that's seen here really nicely, where if you have a BRCA1 either heterozygous or wild-type cells, they're relatively resistant to the PARP inhibitor, but the BRCA1, in this case BRCA2, negative cells are exquisitely sensitive to the PARP inhibition. And there's now, just in the last few months, PARP inhibitors have been approved as a therapy for ovarian cancer, BRCA-like ovarian cancers. And so, our hope now is, because our ability to map out these types of synthetic lethal interactions is so dramatically improved using a type of technology that Max is going to talk about, that we have much better opportunities to identify these types of truly synergistic targets that will allow us to specifically hit mutated tumors and not wild-type cells. So just end with a thought, which is where I started, that thanks to next-gen sequencing and a lot of clever approaches, and both experimental and computational, there's been a huge boon in so-called observational genomics. That is where we see what's mutated, whose genes are changing, what genes going up and down, what epigenetics are changes are occurring and correlating these with interesting biological processes or with important disease and pathologies. That's really a lot of what the GWAS, these genomic studies have been done, or the cancer sequencing studies. And what this does is it generates hypotheses. When these correlations are absolute, like mutations in RAS or P53, you can be pretty sure that they're causative. But in most of the cases, that's not going to be the case. So in the end, you generate a huge number of hypotheses, very few of which you could have tested to sift through the wheat from the chaff. Now, thanks to CRISPR-based approaches, we can change the sequence or modulate the expression of genes, and do this at the same sort of scale that we could do the observational genomics. And it's this intersection, those changes that occur during interesting biological or disease processes that we see by observational genomics, that we know by functional genomics actually impact on the process and aren't just incidental. It's this intersection that's really opening up opportunities for new drug targets and new biology. So that, I'll finish. I took a couple of minutes of your time, but probably give people a bit of a break.